In this episode we're going to look at ecotoxicity. The first 4 or 5 slides are taken from the Alltox website. This first slide gives a definition of ecotoxicity. It reads, ecotoxicity involves the identification of chemical hazards to the environment. Ecotoxicity studies measure the effect of chemicals on fish, wildlife, plants and other wild organisms. Chemical and pesticide manufacturers submit ecotoxicity studies to regulatory authorities to support the registration and/or approval of their products. Testing on animals or plants to determine whether environmental samples such as soil, sediment or effluents contain toxic compounds is also called ecotoxicity testing. Although ecotoxicity in general refers to hazards to both aquatic and terrestrial animals and plants, this brief review will cover only methods for predicting hazards to the aquatic environment. This first slide talks about the animal tests. It reads, the assessment of aquatic toxicity for the classification of chemicals and environmental risk assessments is typically based on toxicity test data for fish, crustacea, daphnids, and algae aquatic plants. Toxicity data from freshwater and marine species are considered equivalent, although this is not true for all substances. The data showing the highest toxicity, that's the lowest acute toxicity values, is to be used in classifying a substance. Fish are used to test for both acute and chronic toxic effects. Think of acute as short-term and chronic as long-term. The paragraph continues, the fish 96-hour LC50 test, that is OECD Test Guideline TG 203, is the standard acute toxicity test. The LC50, that's lethal concentration 50%, refers to the concentration of a test substance that is lethal to 50% of the fish within 96 hours. In practice, this must mean putting, say, 20 fish into a fish tank and putting a gram of cyanide into the fish tank and seeing how many of the fish die. If 50% of the fish die within 96 hours, 1 gram would be your acute toxicity value. In other words, it would be your LC50 value. In continuing the paragraph, it reads, chronic fish tests may start with eggs, embryos or juveniles and last from 7 to more than 200 days. Test endpoints include hatching success, growth, spawning success and survival. This next slide concerns itself with regulatory requirements and test guidelines. It reads, most regulatory authorities still require the submission of in vivo fish lethality test data. OECD deleted the analogous lethality assays for assessing mammalian toxicity LD50 or lethal dose to 50% of the animals in 2002. That's true, they did delete the LD50 lethal dose test for oral toxicity, acute oral toxicity, but they haven't deleted the acute LD50 test for inhalation studies or dermal studies. Anyway, I'm not going to dwell on that particular point now, I'm just going to carry on the paragraph, and the paragraph carries on. In addition to the significant ethical concerns over this type of toxicity testing, most scientists also question the technical and scientific merit of fish lethality assays for determining the environmental impact and fate of chemicals, pesticides and pharmaceuticals released into the aquatic environment. The time and cost of conducting animal experiments for estimating ecotoxicity effects for the many existing and new chemicals are prohibitive. The reason why the costs are prohibitive is that there are so many chemicals to be tested. 30,000 was the last estimate that I heard, and the animal tests take about 3 years. That's 2 years to do the actual animal tests and 1 year to try and interpret the results of the animal test to try and iron out the differences between the animals and the human responses, sometimes called pseudoscience by scientific anti-vivisectionists. These ecotoxicity tests don't suffer from the same species differences of usual animal tests because the results are applicable directly to the environment. So let's say, to keep the maths easy, that they take about 1 year. But if you've got 30,000 chemicals and they take 1 year to do each chemical, obviously that's going to take 30,000 years. So that is what the author of this piece means by saying that it's prohibitive. The paragraph logically continues, to protect the environment and its inhabitants, faster and cheaper alternative methods are needed. In the near term, reduction strategies using tiered testing schemes and/or tests using fish embryos provide the greatest opportunity for reducing the number of fish used for ecotoxicity testing. However, these strategies alone are not sufficient, scientifically, ethically, or economically, for meeting testing needs. Considerable effort is being invested in cellular, genomic, and computational models to meet this need. This slide is about non-animal alternative methods. It reads, cell-based assays, toxicogenomic microassays, and QSAR models are used to predict toxic effects to aquatic organisms. However, current in vitro methods for acute aquatic toxicity are neither standardized nor validated. New strategies for addressing the major limitations of cell-based assays have been proposed. Current in vitro in silico approaches to replacing animals in aquatic toxicity testing include fish cell-based cytotoxicity assays, fish cell-based assays with other mechanisms of toxicity as endpoints, mammalian cell assays using cytotoxicity or other endpoints, bacterial cell assays, usually based on luminescent reporter genes, primarily used for detection, fish embryo assays based on embryo survival and pathophysiological changes, in vitro endocrine disruptor assays, genomic microassays, toxicogenomics, QSAR and other computational programs, and finally test batteries and/or tiered testing schemes incorporating the above types of assays. Before looking at some of the recommendations in the Emerging Research and Methods and Policies article mentioned on this slide, 4 lines down, I'm going to go to the Canadian Council on Animal Care's website just to look at the current state of affairs. Sometimes the Alltox website is a little bit out of date. In front of you, you have a page from the Canadian Council on Animal Care's website, which deals with alternative test methods, validation, and regulatory status. We are going to look at acute aquatic toxicity, which is highlighted in the left-hand corner. The table in front of you, in the left-hand column, you can see conventional test method. As we saw, it's the fish acute toxicity, OECD TG 203. The alternative test is in the second column, and it says upper threshold concentration, step-down approach, a tiered testing strategy to be implemented with OECD TG 203. In the third column, under validation status, it says EU endorsed as a valid testing strategy by the ESAC, which is the European Scientific Advisory Committee, and that's part of ECVAM, I believe. Under regulatory status, it says EU accepted for the assessment of acute aquatic toxicity for hazard classification. And under the column effect or potential effect on animal use, it says reduction reduces the number of fish used by 65%. The other test is the fish embryo toxicity test. Under validation status, it says presently assessing validation. Under regulatory status, it says the OECD is drafting a new test guideline. And under effect or potential effect on animal use, it says relative replacement. This slide gives a little bit more detail about the upper threshold concentration step-down approach, which is a tiered testing strategy. When it says it's a tiered testing strategy, that means the strategy is in stages, usually with in vitro tests at the beginning to work out minimum doses and that kind of thing, and then later stages are actually doing the tests on live animals. This test starts out by looking at the chemical on algae and crustaceans, and you look at the results from this and you work out an upper threshold concentration. You then expose the fish to this upper threshold concentration, and if no fish die, then that is the classification figure. However, if fish do die, then you have to carry on the test, so you go to the next tier, and then you actually undertake the fish acute toxicity test to determine the lethal concentration in fish. So instead of just going straight to the LC50 test, lethal concentration 50 test, you do these initial tests, and that can spare some of the animals, which is 65% of fish in this case. You can see that under effect or potential effect on animal use. If you're wondering whether there's any difference between LD50, lethal dose 50, and LC50, lethal concentration 50, there isn't any difference at all. With LD50, you usually say this if you're looking at acute oral toxicity or acute dermal toxicity. However, if you're looking at inhalation studies, you usually say lethal concentration 50, LC50. I don't know why this is, but I have noticed that. But there isn't actually any difference between the two. There's still a lethal outcome for 50% of the test subjects. This final slide from the Canadian Council on Animal Care's website is about the fish embryo toxicity test. It says newly fertilized zebrafish eggs or certain other species are exposed to a chemical for up to 48 hours, and lethality to the embryos is evaluated after 24 and, if required, and 48 hours. Validation status, it says OECD presently assessing validation. The regulatory status, it's OECD draft new test guideline. An effect or potential effect on animal use, it says replacement. Relative replacement, meaning replacing more sentient animals with animals that current scientific evidence indicates have a lower potential for pain perception. We're now back on the Alltox website under Ecotoxicity, and we're just going to look at 4 more slides. This first slide, entitled Fish Embryos, reads, fish embryos have been found to be a sensitive measure of acute aquatic toxicity. Toxic compounds can impact embryo anatomy and survival. Assays using fish embryos have been particularly good at detecting chemicals that cause developmental delays or pathology. Fish embryo models, while not a true in vitro model, represent one emerging approach to replacing the fish LC50 test. Some consider embryos to be an in vitro test method until the developmental stage, when they can sense pain. And at the end it goes, 4 endpoints for lethality of the fish embryos are evaluated, as we heard earlier, after 24 and 48 hours, and are used as indicators of acute toxicity in fish. If there are signs of delayed toxicity, the test can be extended up to 6 days, 2 days post-hatch. Toxicogenomics was mentioned earlier on in this piece as a possible non-animal alternative method to be used to predict toxic effects in aquatic organisms. This slide gives a little bit more detail about what toxicogenomics actually is. It reads, an extension of the biomarker approach is to evaluate an array of genomics, protein, proteomics, or small molecule, metabolomics, changes in cells, tissues, or organisms. Ankley et al. In 2006 discussed the utility of these types of molecular methods, especially toxicogenomic approaches for regulatory ecotoxicology. Exposure of cells or organisms to toxic substances, or any stressor, results in changes in their normal gene expression pattern. Specific patterns of gene expression can reflect different mechanisms of action of toxicity. The use of toxicogenomics in regulatory toxicology is complicated by many issues, including the fact that other stressors can contribute to changes in gene expression patterns. The vast amount of data generated by experiments and the lack of experience and know-how in translating the genomic data into regulatory decisions. The major benefits identified for investing in toxicogenomics for ecotoxicology decision making are its potential to reduce uncertainty, better science for decision making, and to optimize testing resources, more samples tested faster and cheaper. Toxicogenomics definitely sounds like the future. However, if it is going to be the future as regards regulatory authorities, the people actually working in the regulatory authorities have to be pretty well educated, because this seems to be very, very complex. And at the moment, I wonder whether the regulators will be up to the job of interpreting the data that is put in front of them by the scientists in this particular field. The future complexity of toxicity testing is further emphasized in this slide, entitled QSARs and Other Computational Approaches. It reads, QSARs and other computational approaches will be an important component of any non-animal ecotoxicity test scheme. Villeneuve et al. 2007 developed a graphical model for ecotoxicogenomic research on EDCs, that's endocrine disrupting chemicals, in small fish that incorporates 6 compartments representing the major organs involved in the fish reproductive axis, and depicts the interactions of over 105 proteins and 40 simple molecules, transcriptional regulation of 25 genes, and over 300 different reaction-stroke processes. This model and microarray data from the experiments described by Villeneuve et al. 2007 were used to compare gene expression changes in minnow tissues with the mechanism of action of the endocrine disrupting chemical being tested. I mean, that's really impressive stuff. Very complex, and must be the future in my opinion. I'm highly impressed by this Villeneuve. Anyway, let's see what else it says. The second paragraph reads, QSAR predictions are commonly used for some specific applications in ecotoxicology, especially to fill in data gaps for chemicals with structures similar to ones of known toxicity. The US Environmental Protection Agency makes available Ecotoxicity, a structure-activity relationships program that can be used to predict the acute and sometimes the chronic toxicity of chemicals to fish, invertebrates, algae and other aquatic species. This is the last slide of ecotoxicity, and it's about databases. A bit of a boring subject, but a very important subject, especially if you're a young researcher and you want to compare your results against known results. It reads, databases of toxicity test data are useful in reducing duplication of studies and therefore unnecessary animal testing. The US Environmental Protection Agency's Ecotoxicology database, Ecotox for short, provides toxicity data for 5,900 aquatic and terrestrial species and 8,400 chemicals, and includes information on species, test methods and test results. The US Environmental Protection Agency has also developed the Pesticide Ecotoxicity database, which contains more than 18,000 records for about 805 pesticide active ingredients, compiled primarily from studies conducted by commercial laboratories, but also contains government studies and published data. The US Environmental Protection Agency's Toxic Substance Control Act Test Submission Database is a resource for unpublished technical reports submitted by industry to the EPA, and contains studies on more than 8,000 chemicals, including their environmental effects and environmental fate. The High Production Volume Information System is another EPA database that provides access to health and environmental effects data submitted on HPV chemicals. Other useful databases include INCHEM, Environmental Health Criteria Monographs, and lastly, Hazard Substance Data Bank, HSDB for short.